Thank you very much. First of all, I would like to mention that the major part of this work is done by my colleague Christina Merscher, also from the group of Professor Schäfer. She was responsible for the MRI detection part, but unfortunately she cannot be here today. So I will try to do my best to clarify MRI detection, open points, that may occur. Yes, here's the disclosure. If someone wants to track transplanted or implanted cells, for example, for the treatment of Hirschsprung's disease, these cells are usually need to be labeled before. A common methods in research is the labeling with fluorescence. The labeling is fluorescence-based, and this can be, therefore, primary cells isolated from fluorescence transgenic mice models can be used, or the cells can be labeled with the fluorescence dyes before. A more common clinical approach is the non-invasive tracking of stem cells by MRI when these cells were previously labeled with magnetic nanoparticles before. Therefore, also several techniques can be used. For example, the simple incubation of nanoparticles together with the cells. Transfection agents can be used, even though magnetoelectroporation or sonoporation. And on these pictures here, you can see one example from Michi et al. From the year 2001, where they injected fluorescence-labeled CNS-derived neural stem cells. They were then, 8 weeks later, they were located between the longitudinal and the circular muscle layer. And in vitro, mesenchymal stem cells were labeled with Federick's PLL nanoparticles. These are also magnetic nanoparticles, and this was done by the group. This was done by Frank et al. In the year 2003. So both of these methods are not new, actually. So just a few words for the technical introduction. The particles or the nanoparticles we are using in this, we used in this work are superparamagnetic nano, iron oxide, and fluorescence-labeled nanoparticles. These are embedded in a PLL matrix and functionalized with amino groups at the outside. So what can we do with these nanoparticles? In vitro, we can detect these nanoparticles in two different ways. One way is of course, fluorescence detection. Our nanoparticles are visible or in a microscopic range via a green fluorescence signal, and using the MRI, the nanoparticles are detectable as signal extinction, in the T2 rare sequences. Yes. How do we label, or how was the cells or the neurospheres were labeled in this work? We isolated we started with the isolation of primary myenteric plexus nets, cultivated them, and proliferated them for 4 days, for 5 days to generate the neurospheres. After that, we dissociated the neurospheres for single cells. This was necessary to ensure that the nanoparticles are really incorporated in the cells, in the single cells, and not in between the cells in a neurosphere or something. And after 2 additional days to regenerate our neurospheres, where the cells were labeled, they were embedded in an ECM spot and placed in a petri dish for further observations. Yes. Okay. In our preliminary evaluation, we of course needed to find some working concentration of these nanoparticles, and as you can see in this image here, is that for that when we incubated our single cells from the neurospheres with a concentration of 200 microgram per milliliter in culture medium or 400 microgram per milliliter culture medium, we can detect them in the MRI for several days. However, we decided to use the higher concentration of nanoparticle incubation because when we cross-checked the fluorescence signal, not all of our neurospheres or single cells were labeled or showing a green fluorescence signal. Yeah. What we also did is, because we needed to check if they have some kind of toxic effect or something at this concentration, we did a simple PIs, propylidomidite staining, to ensure that our cells stay alive during this time points. Okay, just to give you a quick overview, what happens when we culture these cells in the pre-named conditions. I put it here, the fluorescence image, the normal right field image, and the MRI image. And directly after embedding the neurospheres, you can see here the neurospheres with a very, very round shape. One day later, these are not consecutive samples, so it's not each day the same spot of ECM. This should be mentioned at this point. Yes. Here you can see one day later the neurospheres are not having this usually round shape anymore, of course, because morphologically differentiation and cell outgrowth from the neurosphere started already. Looking again one day later, neurospheres detached to the bottom of the petri dish or the bottom of the ECM spot and clearly show cell outgrowth. And these outgrowths growing larger and covering a higher or a larger area of space after 3 days in vitro. Okay. It's nice to have these pictures, but nevertheless we need some kind of quantification of our, in this case, consecutive MRI images. So this is at div 0, 1, 2, and 3. This is always the same spot of ECM and therefore also the same neurospheres. To make it for the quantification, we are used to use the grayscale histogram, but to make it more clear, I translated it to the RGB spectrum. What we can conclude from this picture is that the high density of nanoparticles, which are shown here in red, or usually in the MRI images in black, are getting less in number and smaller in size during the days in vitro. Okay. What can we see in this graph here? We can see that the mean area which is covered, the mean area of this ECM spot, of course, which is covered by the particles or the neurospheres in our case, is getting less during the culturing, and the mean gray value of the background increases. This is because the high density nanoparticle high density neurospheres are, of course, outgrowing, and these outgrowths have contribute to the gray value of the background. Okay, we can do something more about that because we are also interested in our neurospheres in this case. Translating the pictures to a binary scale, it is possible to easily count the neurospheres and also to determine the diameter of them. And looking at the mean amount of neurospheres detected in the MRI and their diameter and distributed over the different diameters, we can see that the amount of the neurospheres are getting less during the time of cultivation. Okay, so, but when we want to use our nanoparticle labeled neurospheres, for example, as for the treatment of some ganglionic disorder or some aganglionic area, which is, like for example in the Hirschsprung's disease, it has to be ensured that the neurospheres are all over distributed over this aganglionic area, and therefore the mesenterial perfusion with the particles could be a possible approach. And some of our group members did that before, and they were able to inject fluorescence-labeled single cells via the mesenteric vessels to such a gut segment, and 3 days after that they were able to find the kind of differentiated cells again in the muscle layer, and directly after the injection also in the mucous layer. Okay, what we did to bring our nanoparticle labeled neurospheres in our gut segment, we injected glass capillary, pipette pulled glass capillary, which you usually know from electrophysiological experiments like patch clamping or something, luminally to the mesenteric vessels, and fixated them. And in this image, I hope I can start it by this. You see a time-lapse movie of the injection of contrast metal. I will start it again. In this video, you can clearly see that the contrast solution is distributed over the whole gut segment, and we have no leakage over there. Okay, so we did that. We perfused our neurospheres. We perfused our neurospheres via the mesenteric vessels. The gut segment before showing no black spots in the gut wall, but after the implantation we can find our neurospheres again. Okay, what we did afterwards, we looked at the Hohmann samples of these gut segments, and we can find our nanoparticles again over in fluorescence images until 48 hours after injection or perfusion. Here it's getting at this time point it's getting very difficult because of the incubation of the samples. But in the MRI, and this is why I showed the slide before, we can detect our neurospheres much longer. Yeah, and this brings me to the last slide, the conclusion and the outlook. We can conclude that our enteric neurospheres can be labeled with our superparamagnetic iron oxide nanoparticles. The labeling does not interfere with the neurosphere differentiation. The two weighted rare MRI sequences are a suitable tool for the evaluation of in vitro and in situ experiments. And the labeled enteric neurospheres are detectable in tissue samples for at least 96 hours after perfusion, and the MRI signal is longer detectable than the fluorescence in the case of our nanoparticles. What we need to do Further is, of course, we need to adapt the image analysis or the quantification method for the in situ samples. Okay, so thank you very much. Hope I was in time.